Hi, I'm Callum Edmonds, and today I'll be talking to you about my report, which is titled, What are the rapid effects of steroid hormones on cells of the female reproductive tract? Firstly, I'd like to break down my question and briefly touch upon a few key concepts. The classic model for steroid hormone action works by altering the genomics of the cell, but this mechanism is relatively slow, taking around 20 minutes. More recently, non-genomic receptors have been found to activate second messengers, resulting in rapid changes in cell behaviour, measurable in seconds. Progesterone and estrogen are the two main sex steroid hormones in females. They individually have rapid effects on cells of the female reproductive tract, such as epithelial cells and smooth muscle cells. G-protein estrogen receptors are a type of G-protein coupled receptor that has recently had its non-genomic signaling pathway published. The pathway is very complex. For this example, GPERs are situated intracellularly, but they can also lie in the cell membrane. The main points to take from this are that calcium, SRC and CAMP are all activated by GPERs. The final step is for the MAPK to direct the signaling response through the nucleus and influence transcription factors or genomic estrogen receptors. Once the mechanistic pathway was established, cells of the female reproductive tract were examined. The two studies by Otto et al. Provided a good set of data evidencing that non-genomic GPER-mediated signaling had no significant effect on uterine epithelial cells in rats or on their fertility. Although there was a slight flaw in the fertility study, as you can see with the highlighted data, that the number of offspring was quite low. The average offspring ranges between 8 and 16 in mice, so this may need to be looked at further. A later study provided evidence that GPERs may in fact affect cells of the female reproductive tract. By using a selective agonist, G1, on the receptor, they resulted in uterine epithelium proliferating 3 to 4-fold. As you can see, there is conflicting data regarding the role of GPER as an estrogen receptor in the female reproductive system. Membrane progesterone receptors were identified in smooth muscle cells of the myometrium through immunofluorescent analysis. A signaling pathway was proposed for the contraction and relaxation stages throughout pregnancy. Membrane progesterone receptors are activated significantly more during labour. Once activated, the GI protein initiates a rapid succession of responses, such as these seen here, that enhance the contractile state of the myometrium. Another study examined the rapid effects of progesterone on the ciliated epithelial cells of a woman's fallopian tube and found that progesterone may inhibit cilia beating during ovulation, preventing an egg from passing through to implant into the uterus. There are links here to bovine animal models and some progesterone-only contraceptive pills. Sexually transmitted infections are becoming increasingly more prevalent and have serious consequences on fertility, which is why focus shifted to looking at how estrogen can rapidly affect diseased uterine epithelial cells of the female reproductive tract. When there were no potential pathogens present, estrogen would inhibit chemokine release, yet, as soon as potential pathogens became present, results showed that estrogen doubled in the amount of chemokines. This links nicely to the reproductive cycle. If mating occurs, then chemokines increase due to the increased susceptibility of pathogens. But if mating does not occur, then chemokines are downregulated. Overall, this field is currently fraught with controversy and uncertainty. There is substantial evidence of rapid progesterone and estrogen effects on cells of the female reproductive tract, yet considerable opposition. A gap in our knowledge of the exact structural and mechanistic pathways of these non-genomic receptors must be validated before the physiology can be properly understood. For all this to work in the future, relevant scientists, funding agencies and journal editors need to structurally and financially acknowledge the opportunities of non-genomic steroid hormones on cells of the female reproductive tract.